Hello and welcome everyone to this video about Deep Dive in the Cell Tracking Challenge. Today, we will be exploring some interesting aspects about a fascinating dataset about various cells. A cell is a smallest unit that can live on its own, which has three parts, namely a membrane, nucleus, and the cytoplasm. Here, you can see some of the examples of cells. On the left you can see a three-dimensional visualization of lung cancer cells embedded in a matrigel matrix. On the right, you can see a microscopic image of mouse stem cells. The colors in the image facilitate a better viewing experience. Before exploring some similar examples, let us explore some basic terms first. Segmentation is a technique where the principal goal is to partition an image into regions that are homogeneous with respect to one or more features in the image. Image segmentation is a key building block and is used in many practical applications, including medical image analysis. In this video, we will see various examples of the cell data and the corresponding annotations. To know more about this topic, please stay tuned till the end of this video. In the year 2012, a cell tracking challenge was launched by a steering committee of members around the world, with the aim of encouraging the development of robust cell segmentation and tracking algorithms. Initially, 6 fixed deadline challenge editions were organized as a part of an in-person event. Later on, online submissions were welcomed. To compete in this challenge, the participants need to register online on this website, and only then are they allowed to submit their results online. To get a better idea of the website, let us navigate through it. So, this is the welcome page of the challenge where some basic information regarding the competition is explained. Also, it describes the general outline of how the challenge is conducted and the steps to participate in the same. Under the Participation tab, you can find two options which explain how to register if you want to participate in the challenge and submit the results. On this page, you can also find some information related to how the results are evaluated. The next is the Dataset tab, where you can find description of the dataset. Under the same tab, one can find numerous 2- and 3-dimensional datasets available to download. Furthermore, under Reference Annotations, you can find information regarding how the dataset is structured, and some basic terms are also listed. We will understand these terms later in this video. Delay 1's the results of the challenge are listed here. They are also segregated according to different benchmarks. If you are keen about history of the challenge and organizers, please visit this page. Now, let us look at an overview of the dataset. As a part of this challenge, the participants are given a dataset repository which consists of 2- and 3-dimensional images. To give an example, consider images which show time-lapse sequences of nuclei. Here, one can see images captured at a specific interval and their corresponding images lit up with fluorescent color, which facilitates extra visibility. In some examples, as shown on the right side, you may also find microscopy images of cells moving on a flat substrate. Out of the available data, some of the examples consist of real datasets that were captured through various media, and the other part consists of virtually generated datasets. The virtual datasets can also be looked upon as synthetic datasets, which are generated with the help of augmentation techniques. The datasets are further divided into training and testing data, out of which the reference annotations are available for the training data. We will understand regarding the annotations in detail further in this video. All the datasets, including training data, the corresponding annotations for it, and the testing data are distributed and available in the form of zip archives on the website. Now, as we have understood how the available data is structured, let us have a closer look on the dataset. A plethora of image data is available as a part of the challenge on the website with various representations. Some examples from the 2D data include images for hematopoietic cells or muscle cells of a mouse, which describe about fundamental processes of blood cell development and immune response, or to study cellular process involved in muscle development respectively. They also help the body maintain a stable internal environment. The 3D datasets include images of lung cancer cells infused with green fluorescent protein to facilitate more visualization and analysis to detect whether an individual is infected with the disease or not. Before digging in deep in the dataset, let us have a look on the reference annotations provided in the dataset. The two main categories are segmentation annotations and tracking annotations. The former consists of reference cell masks used to evaluate the segmentation performance of competing methods, and the latter consists of cell markers for evaluating detection and tracking performance. Another way the annotations can be looked at is to distinguish based on their origin. The resulting categories would then be ground truth, which contain the exact reference annotations, gold truth containing human-made annotations, and the silver truth containing computer-generated annotations. For instance, in this image, we can see a real image of captured cells and its corresponding annotations. The top left shows the original image. Second and the third images represent gold tracking and segmentation truth annotations respectively, and the fourth is the silver segmentation truth. The colors in the segmented images, which are generated with the help of masks, show the presence of cells in the ground truth. After knowing about annotations, we will now see a detailed explanation of some examples of the dataset. This dataset contains two-dimensional images of HeLa cells that refer to a line of human cells that are widely used in scientific research and have played a crucial role in various medical advancements. The name HeLa is derived from the initials of Henrietta Lacks, the woman from whom the cells were first collected as a part of her cervical cancer biopsy in the year 1951. These images are from the HeLa dataset, where you can see evolution of cells captured within a specific time acquisition of 10 minutes. The data was captured at Department of Cell Biology in Rotterdam, Netherlands. One of the advantages of this data is that it is widely distributed and used for research purposes. They have been very crucial in various scientific advancements. Also, this is a very versatile dataset as it is used for diverse scientific studies, from virology to cancer research. HeLa cells are known to be robust and can contaminate other cell lines in the laboratory. This could be a disadvantage as it could lead to unreliable research results and complicate the experiments further. This is a typical setup of the equipment used in gathering this data. Here, we can see a microscope Zeiss LSM 510 Meta, having a planapochromat objective lens with a pixel size of 0.19 by 0.19 microns. In this example, we observe three-dimensional images of Chinese hamster ovarian nuclei. These cells are commonly used in biotechnology and pharmaceutical industries for the production of proteins, antibodies, and other biopharmaceuticals. One advantage of CHO cells would be their expression stability. This is crucial for consistent and reproducible production of the antibodies and proteins. A major disadvantage is that the process of developing a stable CHO cell line is time-consuming. The time of acquisition was 9.5 minutes. This data was also recorded at Department of Cell Biology in Rotterdam, Netherlands. To capture these cells, a similar equipment to that of recording 2D data is used. It includes the same microscope, but the voxel size is 0.202 by 0.202 by 1.0. With this configuration of the voxel size of the lens, we can create high-quality and clearer images when compared to other microscopes available in the market. Once clearer images are available as ground truths, the accuracy of the segmentation algorithms would be higher than blurred images. The makers of this challenge have also provided results obtained by various participants. Here, we will highlight the performance of the algorithms and show only the best obtained results when tested against the examples of datasets shown before. This will help us analyze the benchmark results and draw some inference whether the dataset is relevant in terms of the score observed and whether we can use it for real-time use cases. For evaluation purposes, two types of scores are used. First being the tracking score, which assesses how accurately each given object has been identified and followed in successive frames. Secondly, a segmentation score is used which assesses how well the segmented regions match the actual cell or nucleus boundaries by comparing the cell instance segmentation masks. For the HeLa cells, an average tracking score of 0.955 was observed, and when tested it with segmentation benchmark, a score of 0.877 was obtained. Similarly, for the CHO cells, the best obtained scores of tracking and segmentation benchmarks were 0.987 and 0.925 respectively. A lower score here would indicate that either the algorithm does not generalize well or the ground truth data is not consistent. Looking at the results, we can say that the scores from the tracking benchmark have an edge over the scores from the segmentation benchmark in terms of the performance irrespective of whether they were tested against the HeLa cells or CHO cells. Also, such high scores in both the benchmarks indicate that this dataset is very relevant in cases where people have to visualize the path a cell takes over time, which is seen by tracking, or where one needs to study a particular cell and its properties, which is done using segmentation. Talking about the audience of the video, the data and detailed explanation may be useful for cell biologists, professionals working in the area of bioinformatics and medical fields, and students who would like to have an in-depth understanding about these processes and how to analyze them further using segmentation and tracking techniques. Coming to the end, I hope you liked this informative video about the challenge, some examples of the dataset and its use in the real world. Please, do keep exploring about various fantastic datasets.